So continuing with the plasma membrane is a phospholipid bilayer with embedded protein, just like the eukaryotic plasma membrane, and functions in separating bacterial content from its surrounding and selective permeability, just like the plasma membrane of eukaryotes, but has additional functions in the bacteria responsible for ATP production in bacteria, and as well as the photosynthesis in cyanobacteria. So because the bacteria don't have mitochondria or plastids, so the plasma membrane does these things. The photosynthesis happen in membrane foldings called thylakoids or chromatophores. Any damage to the membrane by alcohol, detergents, منظفات like quaternary ammonium, and polymyxin antibiotics نوع من أنواع المضادات الحيوية lead to leakage of bacterial contents and death. Moving on to cell wall, it is a complex, rigid external structure surrounding the plasma membrane. Bacterial cell wall is made of peptidoglycans. This is in comparison to eukaryotic cell walls, which are made of cellulose or chitin in fungi. The peptidoglycans are polysaccharide chains, these ones, that are made of N-acetylglucosamine and N-acetyl-muramic acid, NAG and NAM, cross-linked by peptide chains, these chains. Okay? It functions, the peptidoglycan and the cell wall, it functions in the providing the rigidity, the shape, and protection to the bacteria. It prevents osmotic lysis and increases the ability of causing disease. Some antibiotics, for example the penicillins, work by inhibiting cell wall synthesis, causing osmotic lysis of the bacteria. Now, as you might know, we have two main forms of bacterial cell walls. One that is thick, and the other form of cell walls is thin with additional outer lipid membrane. We use these differences to divide bacteria into two major types: gram-positive, the thick peptidoglycan wall, and gram-negative, which has a thin peptidoglycan and additional outer lipid membrane. By a stain called the gram stain. But some bacteria, like for example the Mycobacterium, have a special cell wall, abundant in mycolic acids, and this makes them stain poorly with a gram stain. We use a different stain called acid-fast stain, and we call these bacteria, for example the Mycobacteria, acid-fast bacteria. There is most of the bacteria has cell walls. An exception for this is one genus of bacteria called Mycoplasma. These don't have cell walls. They have three-layered cell membrane with sterols that prevent osmotic lysis, so they don't need any cell walls. Some bacteria that have cell walls lose their ability to make cell walls. We call them cell wall deficient bacteria, and Archaea have cell walls, but they don't contain peptidoglycan. So this is the difference between gram-positive and gram-negative bacteria. So as we said, the gram-positive bacteria have thick peptidoglycan layer, while the gram-negative bacteria have thin peptidoglycan layer with outer membrane. So the gram-positive bacteria have structures called teichoic acid. These teichoic acids are two types. Those attached to the peptidoglycan layer, we call them wall teichoic acid, and those that attach to the cell membrane, we call them lipo-teichoic acids. The teichoic acids are negatively charged. They attract cations and increase the stability of the cell wall. In a gram-negative, the outer membrane of the gram-negative bacteria have multiple structures. For example, one of the most important structures in outer membrane is the lipopolysaccharide. This is very important virulence factor of gram-negative bacteria, and we will talk about its structures in depth in a second. Other structures in the outer membrane, we have membrane proteins, we have porins that allow materials to pass through the outer membrane, and we have lipoproteins that connect outer membrane to the peptidoglycan layer. The space between the outer membrane and the inner membrane, we call it the periplasm. Okay? Okay, so in gram-positive cell wall, thick peptidoglycan layer have teichoic acids. These are negatively charged and function to provide rigidity and stability by attracting cations. Teichoic acids are two types: wall teichoic acids and lipoteichoic acids. And the gram-negative cell wall, they have thin peptidoglycan layer with outer membrane. The space between the inner membrane and the outer membrane, we call it periplasm. The outer membrane is made of lipopolysaccharide, porins, and phospholipids. The lipoproteins connect the outer membrane to the peptidoglycan layer. Now, the lipopolysaccharide, as we said, is a very important virulence factor to gram-negative bacteria. It's made of two structures. We have the O-polysaccharide antigen. These are polysaccharide side chains that are long and spread outward. So these things, this part is the lipopolysaccharide. The yellow side chain is the O-polysaccharide antigen. This antigen inhibits phagocytosis and protects the bacteria from complement system. Complement system is a type of immune system, and it functions, it attaches to membranes of pathogens and produce pores in that membrane, destroying the bacteria. So what the O-polysaccharide antigen does, it activates the complement away from the membrane. So as we said, it's really long, so it activates it like here and prevent the attachment of the complement to the membrane. So no real threat to the membrane occurs. And also, O-polysaccharide antigen evade antibodies by antigenic variation. So the bacteria can change the structure of the O-polysaccharide side chain, and the antibiotics that were produced to one O-polysaccharide side chain will not be effective to the different variation of the O-polysaccharide side chain. So the second structure in the lipopolysaccharide is called lipid A endotoxin. This component functions after the bacterial death and lysis. So it is released from the membrane, and it is an endotoxin and can cause severe inflammatory response, especially in blood and GI tract. Okay. Now reaching the last layer of the bacterial envelope, which is called the glycocalyx. This is a thick gelatinous layer, mostly polysaccharides, attached to cell walls of some bacteria. So not all bacteria have the glycocalyx, but some of them have it. The glycocalyx is two types. We have the slime layer and the capsules. The slime layer is poorly organized and poorly attached to cell walls. Important example is the Pseudomonas species. These cause severe pneumonia. Capsules are highly organized and firmly attached to cell walls. Important examples of capsulated bacteria are Haemophilus influenza B, Neisseria meningitidis, Streptococcus pneumonia, and Klebsiella pneumonia. Haemophilus influenza doesn't cause influenza, it causes upper respiratory tract infections. Neisseria meningitidis causes meningitis, and Streptococcus pneumonia and Klebsiella pneumonia is a cause for pneumonia. Functions for glycocalyx: it protects against dehydration, detergents, and antibiotics. So it prevents harmful substances from entering the bacteria. It is a source of energy because it's mostly polysaccharides. So when the bacteria needs energy, it can eat the glycocalyx. The slime layer allows attachment to smooth surfaces, for example, the prosthetic devices, and capsules prevent phagocytosis. So this is the stain, how we stain the glycocalyx. The staining is called negative stain because the dye stain the background and stain the bacteria, but not the capsule. So we see a halo around the bacteria.